Thank you all. Thank you for joining us today at the Germ Tech webinar. Today's webinar is our 11th one of the year. I'm Elaine Zhang, the technical support in Germ Tech. We are excited to have Dr. Steve Smith with us today to lead today's webinar on the topic of humanized mouse model choices for autoimmune disease. Steve completed his undergraduate studies at Beloit College before conducting his graduate work at North Carolina State University in the Department of Microbiology, where he studied lipid biochemistry in Baker's East in the lab of Dr. Leo Parks. Steve went on to the Gladstone Institute of Cardiovascular Disease in San Francisco to conduct a postdoctoral fellowship in the lab of Dr. Robert Furries, studying genetically underpinning of cholesterol metabolism. Steve began his industry career at Park Davis Pharmaceuticals, where he studied diabetes and glucose metabolism. Steve joined the Germ Tech as the Director of Business Development, where he assisted with improving Germ Tech's product recognition and expanding our mouse model sales. So without further ado, I think, Steve, you can start off with today's webinar. Steve, the stage is yours.
Great. Thank you so much, Elaine. Thank you very much. Thank all of you for coming today, for joining us on for this presentation today to discuss humanized mouse models for autoimmune disease. So autoimmune disease results from an abnormal immune response in an otherwise healthy individual. Autoimmune disease directly impacts over 24 million Americans. And the global autoimmune disease market is well over $4 billion in the United States in the year 2020. There are well over 80 different autoimmune diseases, with the most common forms being arthritis, asthma, inflammatory bowel disease, lupus, Graves' disease, multiple sclerosis, psoriasis, and type 1 diabetes. Autoimmune disease can be localized to particular tissues or organs, or be more general and systemic. Symptoms often are transitory, including low-grade fever, fatigue, malaise, and just being very, very tired. Autoimmune disease causes are typically unknown, but some may be linked to infection or environmental factors, and some forms are genetic, including lupus. Treatment of autoimmune diseases is typically non-steroidal anti-inflammatories and immunosuppressant drugs. These all improve symptoms, but do not actually cure the disease. Today, we'll discuss disease mechanisms, mouse models for the treatment of autoimmune diseases, and methods for in vivo evaluation of therapeutics using humanized mouse models, studying or looking at models of psoriasis, arthritis, and asthma. I'll begin discussing mechanisms driving psoriasis and mouse models for studying the disease. Psoriasis is caused by dysregulation and complex interaction of the innate and adaptive immune systems, as well as other components such as the epithelium, the vasculature, and cutaneous nervous system. The immune system involvement in psoriasis is widely accepted. The innate immune cells produce key cytokines that activate myeloid dendritic cells, including TNF-alpha, interferon gamma, IL-1 beta, and IL-6. The activated dendritic cells present antigens and secrete mediators, including interleukin 12 and interleukin 23, leading to the differentiation of type 17 and type 1 helper T cells, or Th17 and Th1. cells. Overstimulation of the IL-17 axis with IL-22 and other cytokines can trigger the production of inflammatory mediators, including antimicrobial peptides secreted by keratinocytes. And some of these molecules may function as autoantigens, including double-stranded DNA or RNA. And keratinocytes also secrete pro-inflammatory cytokines, including TNF-alpha, IL-1 beta, and IL-6. Chemokines, CXCL8 through CXCL11, and CCL20. These soluble mediators feed back into the pro-inflammatory disease cycle and shape the inflammatory infiltrate. This leads to the circle of inflammation. The inflammation causes more inflammation. This figure shows the approaches to generate experimental mouse models of psoriasis. Several different procedures may result in chronic inflammation and hyperproliferative skin phenotypes resembling features of psoriasis. Having the transgenic overexpression in keratinocytes is a good model of human psoriasis. Again, overexpression in TGF-alpha, IL-1, IL-6, IL-17. The innate immune system response activated through toll-like receptors, or TLRs, topical application of imigmod results in induction of TLR 7 and 8, which results in inducing dysregulation of the adaptive T cell response and altering the cytokine balance, and then altering the vasculature or nervous function. Topical applications of IMQ in mice has been investigated as a model for psoriasis. Because of its practicality, the IMQ preclinical model has found broad application and is cited in a majority of publications using animal models of psoriasis. So you can see how there's been a dramatic increase in this model being reported and the reports of psoriasis for preclinical models. IMQ is an agonist of toll-like receptors. Topical dermal application of IMQ results in the influx of immune cells and epidermal hyperplasia, and mouse skin is observed to display signs of erythema, scaling, and thickening, which closely resembles a pathogenesis of human psoriasis lesions in overall phenotype and histology. This slide shows the mechanism of pathogenesis of IMQ-induced skin inflammation in the mouse with dramatic increases in many pro-inflammatory cytokines. IMQ-induced skin inflammation is critically dependent on the IL-23, IL-17 axis. Dermal IMQ application induces the expression of IL-23 P19, IL-17A, and IL-17F in the dorsal skin. You can see as measured here. This demonstrates the critical role of IL-23 in IMQ-induced skin inflammation. The involvement of IL-17, IL-23 is observed in human psoriasis, indicating that this mouse accurately models human disease and can be used in the evaluation of therapeutics targeting IL-23, IL-17, and psoriasis treatment. The study was conducted using female BAB-C mice at 8 to 10 weeks of age. Shown here is how we established the IMQ-induced psoriasis mouse model at GEM Pharmatech. This model is initiated following dermal application of IMQ on mice shaved their shaved dorsal skin. Applications of IMQ were done on 8 consecutive days, and mice were treated with dexamethasone or secukinumab as a therapeutic. Secukinumab is an IL-17A antibody that's used in the clinic. This model allows evaluation of drug efficacy for psoriasis treatment. This slide shows greater disease induction following increasing IMQ treatment on mouse skin. The overall histology of IMQ-induced skin inflammation in mice phenotypically resembles human psoriasis pathology. And as shown here, the PASI score was found to increase following the application of IMQ on the shaved dorsal dermis in the animals. PASI is a standard psoriasis area and severity index that allows quantification of disease over time in both humans and animal subjects. This slide shows the overall disease assessment in the IMQ-treated psoriasis mouse model following treatment with dexamethasone and secukinumab. Dexamethasone is a corticosteroid preventing the release of inflammation-inducing cytokines, and secukinumab is an IL-17A specific antibody. And you can see here, again, the PASI scores and the improvement with treatment. GPre-Clinical has developed a number of humanized mouse models for the study of therapeutic intervention in psoriasis. And these with humanized cytokines, transgenic animals, are very good and very accurate models of human disease. GPre-Clinical's K14 human IL-17A mouse model is designed to have conditional overexpression of human IL-17A in keratinocytes. This is a knock-in, ablating the murine version of the IL-17 gene. These knock-in mice display dry, flaking skin by 8 to 9 weeks of age. After reaching maturity, these mice display remarkably consistent skin inflammation. These features show the skin phenotype as observed in dorsal skin of K14 IL-17A mice and littermate controls. Disease pathology is comparable to severe human psoriasis histology, revealing an thickening epidermis, loss of striatum granulosum, and elongation of the papillary dermis, as well as an area of hyper and parakeratinosis and multiple neutrophilic abscesses in the horny layer. I'll now review autoimmune rheumatoid arthritis disease and mouse models recapitulating human rheumatoid arthritis disease. IL-17 producing helper T cells or Th17 cells have stimulatory effects on the osteoclastogenesis and play an important role in the pathogenesis of rheumatoid arthritis through IL-17. IL-17 induces receptors activation of nuclear factor kappa B ligand or RANKL in synovial fibroblasts. Activation of RANKL pathways stimulate osteoclast, promoting bone destruction. IL-17 also activates local inflammation, leading to the upregulation of pro-inflammatory cytokines such as TNF-alpha, IL-1, and IL-6. These cytokines activate osteoclastogenesis, either acting directly on osteoclast precursor cells or by inducing RANKL on synovial fibroblasts, as shown here. Cytokine stimulation of synoviocytes leads to the production of additional chemokines, which recruits leukocytes that amplify the inflammatory response and the production of MMP, which promotes cartilage damage. This slide describes a collagen-induced arthritis mouse model of rheumatoid arthritis. The disease incidence in the preclinical CIA model, or collagen-induced arthritis model, is associated with the expression of specific class II MHC molecules. Different strains of mice have different sensitivities to CIA induction and disease. CIA model is considered the gold standard preclinical CIA model and typically uses the DBA1 model as the source or the model for this disease. This slide describes the collagen antibody-induced arthritis, or CAIA mouse model. Disease in this model is driven by administration of an atherogenic monoclonal antibody cocktail directed against collagen 2, or C2, in mice, followed by the LPS boost at day 3. 3 to 4 days following the initial disease induction, mice begin developing pathology characterized by severe inflammation of all 4 limbs. The disease incidence is 90 to 100%, with symptoms persisting up to several weeks following induction. Macrophage and polymorphonuclear inflammatory cells are found to infiltrate tissue, as shown. And disease is inducible regardless of the MHC class 2 haplotype. So making this, allowing this this model to use other strains of mice other than just DBA1s. And here is a comparison of the 2 primary preclinical rheumatoid arthritis mouse models. While the CAIA mouse model has the greatest and most rapid disease incidence, it is considered too rapid to very accurately represent the chronic human disease as accurately as the CAIA model. Here are shown critical points in the autoimmune disease inflammatory process and where humanized disease mouse models would benefit the exploration of therapeutic interventions. I will show data collected in untreated mice and CIA-induced disease using the humanized IL-17 mouse model. The DBA human IL-17A mice and parental wild-type DBA mice are found to have very similar populations of B and T cells seen in both male and female mice. The CD4 positive and CD8 positive T cell subpopulations in both male and female DBA human IL-17A mice are similar to those subpopulations observed in the wild type DBA mice. The NK cell subpopulations measured in wild type DBA mice and DBA humanized IL-17A mice in both males and females were also found to be quite similar. These findings indicate that the overall immune system in DBA human IL-17A mice, knock-in mice, is quite similar to the wild-type DBA mice, other than the expression of the human IL-17, indicating that this is a very good model for or mouse model strain for the study of diseases such as rheumatoid arthritis. Here we see the DBA human IL-17A mouse strain used in the CIA arthritis model. This strain shows clear improvement of disease pathology when treated with secukinumab and an unnamed test article when compared to vehicle treatment. You can see here. Then also this histology, the serum, cytokine levels, and pathology score were assessed. These results show the utility of this DBA human IL-17A mouse model for discovery and development of therapeutic entities against rheumatoid arthritis pathology. I'll now review autoimmune asthma disease and mouse models recapitulating the human disease. The primary feature of asthma is airway obstruction, which is caused by reducing the airway diameter. Narrowing of the airways is mediated by chronic inflammation of the airway wall characterized by the infiltration and activation of immune cells, including dendritic cells, eosinophils, neutrophils, lymphocytes, innate lymphoid cells, and mast cells. Upon recognizing allergens, allergen-specific Th2 cells produce type 2 cytokines IL-4, 5, and 9, and 13, leading to the accumulation of eosinophils in the airway wall, mucus overproduction, and synthesis of IgE by allergen-specific B cells. After Th2 cell generation and lung draining lymph nodes, the subpopulation will interact locally with B cells. In mice, the allergen exposure is able to trigger the production of IL-33 thymic stromal lymphopoietin, or TSLP, and IL-25 by ECs. All these epithelial-derived cytokines contribute to the establishment of Th2 response in murine models of eosinophilic asthma. There are several mouse models of asthma, each with strengths and weaknesses in recapitulating the human disease. Shown here, the OVA-induced acute asthma model, or OVA. This uses a subcutaneous administration of OVA followed by intranasal OVA challenge that drives the disease. Advantages are that this is an eosinophil-driven inflammation and the OVA-specific IgEs that are generated. Disadvantages are that it's an artificial antigen and the immunization route being sub-Q initially, and it's a very rapid disease study. The house dust mite induced acute asthma model uses an intranasal administration of the house dust mite, or HDM, to induce disease. Advantages are that this is an allergen and induction route that is very relevant to human disease. Disadvantages that the allergen-specific IgE in mice is not very robust, and it's a very rapid, also a very rapid study, even more rapid than the OVA study. Alternaria extract-induced model. Oops. Oh, there we go. Alternaria extract-induced model of disease uses fungal Alternaria extract intranasal administration to induce disease. The advantages of this are that it's a fungal allergen that's relevant to human disease and induces innate lymphoid type 2 cells. Disadvantages is that it's also a very rapid study, and it may not accurately fully represent the human disease because of its rapid onset. The combination of house dust mite, ragweed, and aspergillus intranasal challenge is a better model in that it's very relevant allergens driving human disease, the innate lymphoid type 2 cell response, disease pathology remains for months following induction, and it represents the human chronic allergen disease. So GPre-Clinical has developed a panel of humanized mouse models of the Balb C strain background that can be used to model human asthma. Shown here that there are marketed therapeutics for asthma that focus on multiple proteins in the disease cascade, and GPre-Clinical has models for each of these steps along the way. Depending on the humanized genes and proteins, models reflect allergic, eosinophilic, neutrophilic asthma that are either type 2 driven or non-type 2, non-T2 driven inflammation. And shown here are models fitting into each of these categories. This slide shows humanized mouse models for autoimmune disease research and therapeutic discovery efforts that have been generated by GPre-Clinical. The strains highlighted in red are strains of mice that are currently available from live colonies. The other strains in black are currently maintained as frozen sperm. These additional strains can be rapidly reanimated for investigators wanting to use our models for their research studies. In addition to the models I've discussed in today's presentation, we've launched the Humanized Mouse Model project with the goal to generate over 700 mouse models with humanized genes for use in drug efficacy and safety evaluation and disease models. Many of these models are applicable for the study of and therapeutic development to address immuno-oncology, neurodegenerative diseases, metabolic diseases, inflammatory diseases, and autoimmune diseases. GEM-Pharmatech is open for collaboration with our customers to validate these disease models and develop additional models. Thank you all for your interest and attention today. I'm very happy to answer questions regarding our mouse models for autoimmune disease. Thanks.
Thanks, Steve. Thank you for your wonderful presentation. If all of you have any questions, please type in your questions in the Q&A region. We are more than glad to help and discuss here. Let me check if any questions from the audience. Hi, Steve. Here I do receive a question from our audience that, as we know, B6 mice is the commonly used strain for gene editing, but why your humanized model for asthma study are generated on biopsy background? So here is the reason we received from the audience. Do you have, Steve?
Thank you very much for the question. We know that allergic asthma is associated with T helper 2 or Th2 cell responses. Upon allergen recognition, allergen-specific Th2 cells produce type 2 cytokines, leading to the accumulation of high numbers of eosinophils in the airway wall, mucus overproduction, and synthesis of immunoglobulin E or IgE by allergic-specific B cells. And the BALB/C mice tend towards the Th2 predominant immunological response. Well, the C57 Black 6, which is very commonly used for other studies, has a Th1 type bias to its pathogen response. So the most commonly used, so again, that's the most commonly used in other studies. BALB/C has been used for these studies where Th2 response is very important. GEM-Pharmatech can directly conduct the gene editing and make a gene knockout or transgenic in any strain background. And again, we've been looking at the best strain background, whether it's a Th1, Th2, or other strains for all disease models. Thank you.
Thank you, Steve. Here I received another question about the cytokine humanized mice. Audience wonder if you just express human cytokine in cytokinase in mice, will it be a problem for the human ligand and the mouse receptor recognition? Did you do any tests?
Interesting question. We'll consider the question as we create humanized mice on a case-by-case basis. Again, we're looking at, we have a goal of creating over 700 mouse lines with carrying humanized proteins or expressing humanized genes. And depending on the disease focus, whether it's neurodegenerative, metabolic, immune-related, we'll use the appropriate background for that disease. For some cases, we humanize both the ligand and the receptors, like the TSLP receptor humanized mice. In some cases, the single humanized mice will also work for drug evaluation, such as that DBA human IL-17A mice can be used for, and then collagen can induce arthritis in humanized IL-17 mice, And drugs targeting the human IL-17A can reduce arthritis. So that's an indication right there that it can be utilized using the DBA background.
Okay, thank you, Steve. So here is the last question I need to forward to you. For the IL-23 and IL-17 analysis part, one of our audience asked, where did you collect the sample? Where did you collect the sample? Can you help?
It was detected, samples were collected in RNA level from tissue blocks and from the skin, primarily the skin. Since we're looking at psoriasis, IMQ was applied on the skin, so the cytokines dysregulation happens locally, but not systemically. We did look at serum cytokine levels, but we're focused on the skin because that's where the disease is focused in those animals. It might be difficult to detect the increased IL-17/23 in the serum after application, so we were really focused on the very specific, very targeted, close areas.
Okay, thank you, Steve. Thank you all the audience for your time. If you have any further questions or any demands or needs about our humanized mouse models, you are welcome to email us to the sales@genpharmtech.us. We are more than glad to communicate with you about more details about all the models we have. I think that's all for today's webinar. Thank you for all the audience for your time. Thank you, Steve.
Great. Thank you very much.
Okay. Goodbye. Goodbye, everyone.
Bye.
Looking forward to see you soon. Bye.
Yes. Bye.